Yeah, share screen, not window, share screen. Yeah. I mean, I don't need it at all right now, but
Also small pen, right? Yeah. So there's no presentation, right?
No, there is nothing.
Okay. Now we're ready to present.
Yes.
Yeah, it's okay. So,
Cool. Okay.
right, all set.
Yes. Hi, so I'm guessing almost everyone is here. Yeah. Okay. So again, please download these two software. That's what we'll be using. How many of y'all are done downloading the software? Please. Okay, fine. No, as in, oh, okay.
This is Athulya. She's a last year PhD student.
Yes.
The last years. Done a lot of work with phylogenies. So she's going to do a tutorial where go through and do some work with building phylogenies and making inferences based on that. So welcome Athulya and thank you.
Yeah.
Is it just that one?
Can you go back? And can you just type mega and mega phylogeny? Can you just type that?
That one? Okay,
Yeah.
perfect. And the fig tree is this one?
Yeah.
Yep.
Oh yeah, there is macOS. No problem. Yeah, yeah. Right, but a lot of people haven't downloaded the software yet. Yeah. Yes. No, you need both. So there's going to be 4 sessions. The first is downloading sequence data. The second is aligning them. Third is just a demo on how to partition your data sets and how to set up a phylogeny run. And the fourth is again a hands-on session where you would be working with the tree output that you get from the phylogeny that you run. But we'll be working with example data sets which you get along with fig tree. So you just need to download the software. The folder will already have the phylogeny that we'll be looking at. You can call, yeah. You should get
So
dot zip. No, where it's here itself. Okay. Yeah, for Mac OS you do use dot zip files. Like,
Yeah,
then
which
get the
do not
dot
zip,
zip.
yeah.
Yeah.
Hmm.
This looks complicated. Yeah, sure. So the dot zip. This is a Windows system, right? Then dot zip. Can you extract the files? Wait 1 minute. Okay. Cool. Now there's a Fig3 folder which is not zipped. But why does it look like this? Hmm. Okay, fine. It's fine. If you run the .exe, it's working. So
Or should it just be?
like they are more or less instantaneous. It's showing 1 hour. Oh, that it's showing 1 hour to download mega. Now, how much time did it take? Like just okay. Hmm. Yeah. Okay. So for the first session of downloading sequences, we don't really need any of these software. So I guess we'll get started. For the next session, I mean, for the alignment is when we'd be needing mega. So you can like set up your downloads until then, I guess, and it'll hopefully be done in half an hour. Okay. So we are going to be downloading sequences from this online repository, which has all kinds of sequence data from published work called NCBI. And we'll just be looking at some of the ways with which you can retrieve data, which makes your life easy. But then there are so many options on NCBI. I'm sure I've not even seen half of them, which you can explore later by yourself, I guess. Okay. So I'll be doing it here on my screen and you can just mirror it, I guess. So there are all these different data sets that is on NCBI. We are going to be looking for. So the whole session is only for nucleotide sequence data, but the ideas that or like the concepts would be similar regardless of whether you use morphological data or protein data, for instance, but I guess the softwares would be different. Okay, so we start with choosing the data set that we are interested in, which here it is nucleotides, and then here you can search for whatever the species or the group that you're interested in. You can try out whatever species or something that you guys work on, for instance, and then space. We'll also search for the gene that we are looking for. For instance, if I am doing this. So Papilio is a genus of butterflies. And wingless is a gene that is commonly very standardly used in phylogenetics in Lepidoptera. So if I search for this, you get all these Papilio sequences. So, and you can select multiple sequences at the same time. And then you download it. So the format you choose is FASTA, which is one of the most common for formats for sequence files. It's basically just a text file which you can also open on Notepad or Notepad++, for instance, and look at what it's like. So if you just say format is FASTA and create file. Yeah. Oh, I selected some random 4, but that's okay. You can just try out whatever. Yes, this is all the wingless gene for Papilio. But then you can also, for fun, like try out some other, organism or some other gene if you want. No, you can also just search Papilio and it'll just Papilio or just the name of the organism, and it'll show you all the genes there are. And there's also ways where you can, there's a taxonomy option where you can just go to the organism itself and then it'll show you all the genes organized for the organism. So, but when you're downloading one sequence at a time, or usually you would have an idea of what is the data set that you're aiming for. So you will already know what the sequence name is. Okay. So if I create file, so we're just going to look at what a FASTA file looks like. Yeah. Yeah. But now what?
Whatever you want, you can.
As in the whole screen is shared, but that's not what you wanted. Okay. Okay, so this is what a FASTA file would look like, which is a text file. It starts with like a greater than symbol followed by the description of which has no format really. It can be whatever. You can have where it was collected from and the name of the organism, whatever. You can add whatever it is that you like, and then followed by the sequences in the next line. So this is a list of sequences. So you have multiple different sequences that's arranged like this. You can edit sequences just using your Notepad file. You can delete 2 of these sequences, and when you open them in Mega, for instance, which is something with which you can analyze the sequences, you would see those changes reflected there. So it's basically just text files is what I'm trying to say. Okay, so using the sequence ID is another way in which you can download these sequences. For instance, all of these sequences, they have, as you can see here, there's an accession number which is unique to them. So in case you know the accession number, for instance, you're looking for a particular sequence that you saw in a paper and you need to work with just that one sequence, or you need to see what its similarity is to the sequence that you have. So you can also download, you can just type in the accession number here and download sequences, the exact sequence that you want. 1 minute. These are just some examples that I had kept ready. So it takes you directly to the sequence that it opens the sequence, basically. It doesn't show the list anymore. And there are all the details regarding the publication here and the coding sequence and the sequence itself. Yeah, how I did that. Okay, 1 minute. You can search for this ID, I guess.
So if you have a range, you can define the range here as well.
Not here. That's the next thing. There's this utility called NTRES, where you can provide a text file which has the entire range, and it will just give you all the sequences in this range. So you just need to have the text file prepared, and how that works is, okay. So you go home. Then you go to all resources. And this thing is called batch entries. Where is it? No, tools. Oh God. Oops. Batch entries. So we're again saying the database is nucleotide and you're choosing the file. If you say retrieve, there it says there are 5 unique sequences, and then you just get those 5. Okay, so I'm going to leave this here. And why don't you all try making a text file and retrieving these sequences, is because this is what we are going to be aligning in the next step. You can do them, I mean, yeah. Yeah, in the tools. Batch entries is the thing. Yeah. Yeah. This is gonna be, can't read it. I'm sorry. Yeah. As in, did you download all these 5 sequences? You got the sequence list, right? That's ready. Now we will try and align them next. So that's it on Mega. Yeah. So you just change the database to whole genome here. So until now we were only searching in the nucleotide database, but then there is genome, or I think there's transcriptome, protein data, there is all sorts of things. So if you say genome and again do the same thing. Okay. We'll get all the whole genomes there is for papillio. Yeah. Can I see it? Because you go there, as in you go to batch, no, go to tools and batch entries, you go there and then there is upload file.
Athulya, maybe just show how to export the source once again.
Okay. So all resources, then tools, and then you have batch entries here. Yeah. The complete record. Yeah. Oh.
One question. So these are my sequence. I have taken this 4 sequence of them. And I have to choose the file from here. So
Yeah. So you downloaded those 4 sequences, just selecting them.
yeah.
So did you, to do the batch entries thing, you need a text file with just the accession numbers that I had put up, for which I can put up again.
Okay, sure.
I mean, I'm sure even if you try aligning these sequences, it's going to be the same thing, but just so that everybody aligns.
Yeah, notepad, I got and then Madhava, yeah, now
To individual people, I'd switch it off. I shouldn't. It's fine now, right? Yeah.
Oh, it's saved as a Word doc.
Mm, no, it's like the mega format actually.
Oh, okay. In which way? Is it okay?
You can open it directly also, say align.
It opens on this.
Yeah, you can also right click and open using Notepad or Notepad plus plus or something.
This was only individual. I don't know where it's open here.
So this is what it would look like. It's only one. I thought we selected three.
This
This is the first one. Okay.
Now it was open.
In file open. Open the file session and then choose the sequence. And then say,
It's both align and
it's the same thing. Say, no, say align. Second time is taking slightly longer, but
large.
seems small. Okay, but yeah, then it should open up. It didn't open. Okay, 1 minute. It must have. Okay. This is weird. 1 minute. We'll try. But see, this is not how you wanted it. Okay. Did you download Mega just now?
Yes, today.
But I don't know why this is happening. Okay, try some. I'll be back in a minute. Let's see what's happening there. Yeah.
Java download नहीं थे इसलिए run नहीं करा.
What needed Java? Mega. हाँ ठीक है. Yeah. Yeah. Okay. Yeah.
This I have run, and then batch entrance I've selected this.
No, you don't. Wait, what is sequence.text? No, this is not. So the input that needs to go into entrance is that. Just the sequence. Yes. So it's another way to get sequences at the same time. It's not to get higher. You understand? No?
Yeah.
Okay. Okay. Okay, there was this one last thing that I wanted to show is if you're looking for mitochondrial genes and you can't find the specific gene that you're looking for, you can also look for the whole mitochondrial genome. And then if it's been annotated, which means that you know from which base to which base is what gene, then you can just also get the coding sequences of the gene that you are interested in. And again, you have the sequences, you copy paste it, or you get the FASTA, and then you can work with it. So that's just one last thing. Now, did everyone get at least some sequences they can align in the next step? Okay, and does everyone have mega by now? Okay, cool. Then that's all about retrieving sequences. Now let's align them. This is how it should look like when you open the sequences in Mega, where you go to just file and open the sequence file. This is what it should look like. With maybe small differences because mine is not the latest version of Mega, but more or less like this. Ah. Has everyone got their sequences open? Okay. So just select all of it and then say align. And then there are different algorithms that Mega provides. So Mega is just, you can do multiple phylogenetic analysis. You can even reconstruct a tree using Mega, but alignment is one of the things that you can do using it. And that's what it's most commonly used for. So there are different algorithms that they provide. So what I commonly use is the muscle algorithm. And then you just say align by muscle. And you don't really need to change any of these parameters for any basic analysis. You can just use the default parameters and it should work fine for most cases. And just say okay. Oh, you're looking for a specific.
So I've just chosen that one.
Oh, wait, I think a lot of people got this confused. So the input that goes into NCBS is a text file that has the list of accession numbers that you want. So it's just another way of getting a whole range of sequences in a single go, and which is very convenient because most papers would have the range of the accession numbers in their materials. So you can just get the whole group. So it's not the sequence that goes in, it's the accession number. And if you have a bunch, it's the text file with that goes in as the input. Yeah. So it might not look exactly like this because now everyone has probably like different sequences, but yours still is like doing weird things. Oh no.
So now I could get this as in the notepad.
Oh, 1 minute. Can I see your sequence file?
Yeah. It's called
This.
Yeah.
So, can you? Oh no, that's not how it works for your. Can you make it a .fasta file? I think when you downloaded the sequence file, I'm not sure what the format it was that you chose. Did you choose FASTA? Okay, but anyway, like there's no dot here or something weird has happened. So call it Hydra FASTA .fasta, I guess.
Okay, I'll rename it.
Because now it's a text file and not a FASTA file.
.fasta. Now I'll try it here.
Even though
Not showing.
it maybe we like changed where it was with all this changing. Then just open this one. This is a FASTA file. It says already, so it should work. Align.
Just close it.
Okay, go to the downloads and downloads. And directly just double click it. Oh, not this. That's a mega sequence thing. The FASTA file one. Can you just double click it?
Does it happen often? No.
Not so much. But then this is some small problem which we just can't figure out. It's open, I guess. Okay. Finally. Okay. So these are the Hydra things that you did want, right? Okay. Yeah. I don't know how I'm gonna do that. Just control it.
So some math problems I don't know.
Oh, okay. Then just go straight ahead to align. And then choose muscle. And then it will ask you.
Yeah, select all.
She didn't ask her, but say okay. This is the aligned one, right? I mean, so usually the pop-up message comes up saying you've not selected anything. Do you want to select all of it? It showed up for him. I don't know why it didn't show up for you, but. Yes. That's because that is a slightly more complicated thing happening there, which I will show once we are done with.
Why? Because I align according to like sequence are same.
That's because to show you all something, I have changed some stuff in the sequences there. So that's why.
So I just saved it.
Hmm.
Is this right?
Hmm.
I deleted the point ones because it wasn't working earlier, but yeah, and that happens. It's just the
Is there anything
TXT
in the
file.
file name that she has used or
Hmm.
something that she hasn't included, which is preventing this?
That's
Can I see
file.
the file
Yeah.
thing? It's a text file, so that's all that should matter.
I just copied these from like those codes. The locus.
So at times, like when you're using a Linux system, there are things that when you copy that are there, but you can't see. So I'm sure if you rewrite this yourself, like type it, I'm sure it'll work. But
Copy paste should work.
no, at times,
Text file.
yeah, but then sometimes, at least with Linux, it happens a lot where they say there is some character which is not visible, but which is there. I don't know what exactly it is, but if you type it out, then it works. Just type 2, I guess. No, at least 3, so that you have 3 sequences to align.
Well, should happen. Text file does not show characters which things. Text files, especially after copies. Maybe check this
Why that happens.
I'm sure.
Yeah,
The alignment that I have open right now is
it's very bad.
is this
Yes, that's not what it would be like, yeah. So I wanted to show some things that could happen with alignments. So with one of the sequence, I have changed something. So that's why it looks very bad and different there. Yeah. Which is good. I mean, there could be gaps also. It's not that gaps are bad, but these are different species of the same genus and it's a pretty conserved gene, and to have this kind of a pattern for a gene that is expected to be more or less similar is weird. So this should put you off saying there's something wrong with the alignment that I have like created. But not that gaps are in general bad.
Catch after this, like I just downloaded it and then I imported this file. Next,
But
I
then
have to
it's
align.
opened as some.
I have to save this.
Close this. Oh, this is just the. Okay, sorry.
That's the sequences.
Ha. Okay, close it and then go to Mega. No, open just the Mega app, like search from here, just open it. Is this how it's opening? Ha. Then file, open a file session, then choose your sequence, which is a text document. It should be a FASTA file.
Okay.
You can just rename it and type .fasta. That's probably enough. Can you again show me the, like with her, I think yours is, it's again showing edit text. So when you do the send to thing, do you choose what format do you choose?
Plain text and faster nucleotide.
Okay, that should be fine. Okay, can you just rename it at .fasta though? Like it's now a .txt. Did that work for you?
No. I have to re-download it.
Do most people have their alignments done? We'll look at what's happening in that alignment in a moment, and then we'll go to the next step.
Re-download.
Just rename it and call it something .fasta. The same thing.
Now getting downloaded as text only. Even though it's,
Cancel it. Cancel it, right? Send to complete record file. Wait, then
doing
how did
only
you
the
do?
coding sequence? Choose this second one.
Oh, okay.
Okay, complete it.
Okay, so choose the complete record or not just the coding sequences. Even though I would have not assumed that would be a big problem, it apparently
Now
is.
it is fast.
Okay, so that's all. Yeah.
Four years are like 391 base pairs, but the
Hmm.
first one is 389.
Hmm.
So what it's aligned from the start, right?
Hmm.
So how is it decided, like align, why do you align the start one and like, two gaps here? Right?
So that depends on where those nucleotides are missing, right? So the process of alignment is to make sure that you're only comparing regions that are homologous, which means they have the shared evolutionary history. So let's say this particular gene is 400 base pairs long, and you have 3 sequences that are exactly 400. It starts with the starting codon and ends with the stop codon, but one of them has 2 nucleotides missing. So when it's aligning, if the missing nucleotides are at the end of the gene, the aligning software should ideally figure it out that the gaps should be at the end, right? So it's not that it's just starting from the beginning. Depending on where the missing nucleotides are, it would align appropriately. At the end, it should just be that all the homologous regions are aligned together. That is the goal.
Cluster hypothesis.
You can try cluster and see what happens. So these are 2 different algorithms that have some the basic idea might be different, like the maths that you use might be different, and they'll all have these pros and cons generally. Okay, yes. But both of them have been known to be pretty decent. So you should ideally get the same result, even if you use cluster, considering this is just a small gene with just 4 samples. So even if there are their strengths and weaknesses they do have, they should not show for such a small data set. They should all work pretty much the same is what I'd say. Did you try cluster? Is that what happened? Yeah. Okay, so somebody was asking what would be the difference when you tried aligning using cluster instead of muscle, which is just another algorithm for aligning sequences. So you can try that. And I was saying how they have slightly different algorithms on how they calculate the alignment should be. And they have their different pros and cons. But considering this is a very small data set that should not be reflected in the alignment that you see, they should be identical actually if you use cluster or muscle or others, like there is MAFT, for instance, and T-Coffee. There are other algorithms as well that's not provided here in Mega, but they all have different strengths. For instance, MAFT is supposed to be good when you have lots of insertions and deletions. Like if this is how the real alignment is supposed to be, they're really divergent sequences. They're not closely related at all. There are actually lots of gaps because in some sequences, there have been lots of insertions, for instance. So muscle might not be the perfect software for that. That might be MAFT, the algorithm for that. MAFT would apparently handles indels better. So they have their strengths and weaknesses, and depending on your data set or how related they are, you can choose them. But for most data sets, there should not be a huge variation because these are all still currently and very popularly used. Okay, so almost everyone has their alignment done, right? But they all look cleaner and nicer, and you have all the columns with the very similar bases. But here you can see that there is something really off. These are really closely related species, and you can see that the top 4 are mostly similar, but the last one seems to be what is off. So if you see, when you download sequences, there is always a chance that what you thought you've downloaded is not really what it is, because you're getting data from other people's work, and there could have been errors at some point, at any point. So it's also possible that this itself is like a wrong sequence. It's not wingless of a papilio. But one thing that you should try is that it's possible that just this one sequence, it was uploaded as a reverse complement. So there are some not software, the algorithms, which automatically reverse complement a sequence to see if it then aligns better, but muscle does not do that. So you open the sequences again. You can't unalign them once they've been aligned. You have to open them again. And then the last one was the one which was very different. So you right click and reverse complement it. Okay. Then you select all and try aligning again. Which has solved the problem. Okay. So you can also, if it's a protein coding gene, you can also try translating it to make sure that it has been aligned properly. You can also try that with your alignment now. You should not ideally see many stop codons in the middle. Ideally, you should not. You should only have amino acids, but there is a possibility that you have partial sequences of the gene, and this one really doesn't start from the first base of a codon. So you can also try deleting an entire column at the beginning. I mean, I don't know, when you all translated, did you get any stop codons? 1 minute. No, that's not what it has done. Yeah. Oh, the asterisk. Yeah, so when I deleted the first 2 columns, which sort of removed the offset, the extra nucleotides that were there. Now you see that when you translated that they properly align, so we have more confidence in this alignment that this is how it should exactly be. Somebody wanted me. Yeah, you guys wanted me, no?
No, this is.
Okay,
I'm
I thought
just
somebody
telling-
raised the hand. Huh?
Like I was telling, I have got stop codon, so I'll just remove it from here. So the stage shifted out.
Wait. How did you remove? What did you remove?
Just like this. It shows a stop codon, I guess. No, sequences are different.
Okay. Okay. But then you don't just delete the column that shows the stop codon. You go and see if this is a pretty long sequence. So it's possible that considering all of them have a stop codon there, that is what it's supposed to be. It is likely. But if they have randomly placed stop codons, you delete nucleotides from the beginning of the alignment. It's because in case it really, you know how there are 3, they should be ideally multiples of 3, but maybe some of them aren't. And that's why it's being translated weirdly. So if you delete 1 or 2 column from the beginning, if that was the problem, then you should not have these random stop codons, right? um,
Like
it's the
I'm
small
just trying to-
sequences, no? I don't know.
I don't know. It's taking so much time. I cancelled and retried sequence.
Different sequence and trying.
1 minute.
Huh.
I don't know.
I have another sequences.
Yeah, are you using the Papillio ones or did you? Wingless
Wingless
only.
only.
But
Whole genome should-
wait. So this is Bombyx mori, which is a moth.
Okay.
I don't know why you're. Oh, wait.
Papilla.
Ha. Papillio. I mean, it's fine because it's taking a long time because I think these are 3 very random sequences and it's trying to align it. And that's why it's taking such a long time. So that is the only thing. It's not that you did anything wrong, but then it's just that these sequences are not similar at all. That's,
I'll just take like similar sequence.
yeah. So for the next step, which is mostly a demo, we'll not be using this. So did typing it out work? What?
Just went to Mega and
Okay.
characters on.
Okay. Okay.
Yeah, I didn't focus on the off one. Which is why the visualizing.
What's happening?
She's getting a lot of stop codons and I don't understand how.
Okay, so did you try deleting the columns as in?
No,
You haven't done anything, right? So it is possible that you got the sequences that you've selected, they're not really multiples of 3. So it might not really be starting from a proper codon. There might be 1 or 2.
Go to the downloaded once again and see what you have downloaded.
But you can also, to see if that's what's happening, for instance, you can try doing this thing where, but
I
it looks,
suspect there's something wrong. See, this one
Hmm.
has so many gaps. Or it's just
Oh.
a smaller one, is it?
Yeah, it's probably, yeah.
Or just. a
Oh, she's
smaller-
used cytochrome, CO1, so it'll be, okay. Oh, wait, you have used CO1. Yes. So when you translate, the mitochondrial, the codons are differently used for the mitochondrial genes versus the nuclear genes. So there is a genetic code which is different for, you should say that the current genetic code is standard, which is what is for all the nuclear genes. Since yours is a mitochondrial gene, you should say it's not. And then you should say it's invertebrate mitochondrial. Yeah. And there are all these different codes that are supposedly like different. So
Okay.
now, even if now there are, then you should try deleting what I said. The first column you delete first.
Let's see what she has downloaded also.
Okay.
I'm not sure what she has done, how she has downloaded. Otherwise, you shouldn't be getting so many.
Ideally not, because I chose these because they're very
She's also
curated.
choosing,
Cluster.
some different settings, perhaps just like you did with the genetic code.
Just select them and
Oh, no, not the stop codons. So
From the file.
no, you just go back to the DNA sequences, and we're saying that it's been aligned, but then there is a possibility that there are 1 or 2 extra nucleotides in some of these sequences. So that's why they're not in frame, or they're not being translated in units of 3. So that's why something random could be happening. But then if you delete, where is delete? Oh, sorry. And then you say yes. There's still, we only deleted 1, right? 1 column until now. So we can also try deleting 1 more. Yes.
So basically, if the codons are not in line, they're not starting with the first position, then you will have this problem.
There. So that was the problem. You had 2 extra nucleotides in all your sequences. Okay, so also
Starting position.
the 80G,
sequences,
but
which case you don't have to keep deleting.
no, you're saying while you're downloading,
You start a line.
not on Mega.
So happening in a sequence, we have the first place in our sequence was
Yeah. And it was not even a unit of 3, so it was randomly translating. But it's difficult to actually get sequences from the starting codon always, because you're downloading them and you can only download what is out there.
Why this is not opening?
you will have to do a line.
Oh wait, it's wingless.fasta, open with Mega 64.
Even normal one, I'm unable to. Something is wrong.
Wait, so you've not opened Mega at all until now. Okay, 1 minute. Let's try opening it another way. What is it? I forgot. Open it.
So she had also downloaded a micro.
Did you down? Can I see your download again?
Faster.
Because there's already first it says .fasta.txt, so it's not a FASTA file currently. It's a text file. Rename it. But it says .fasta here, no?
I mean, I just changed it to .fasta. That's it.
But oh, that is only. Can I see the download that you have? Because
I did it here.
can I see the tab where you have downloaded it? Like the NCBI tab, you've closed it.
Yes.
Oh, okay. It's better if you just choose the format as FASTA there itself instead of changing it here. Okay. Next to a smaller. Like the demo would be fast. So
So one question here is it okay if a stop codon pops up in your data set? And it's not okay if you're going to do all the downstream analysis. Because after this, Athulya will go over this, but you are then going to assume some model of molecular distribution. You have to define some parameters. So if your alignment is not very good, then your everything downstream is not very good. So especially for inexperienced labs and people or if something doesn't make sense. Reviewers who know the field, one of the first things they will ask is for your alignment. So alignment seems like a very, very simple thing, right? You just download sequences and you align it. Mega does a pretty good job. But the thing is, if you mess up something there, then everything downstream is going to be wrong. Simply because of all these steps, depending on your basic alignment data. And we are seeing there are stories of how an experienced PI did not pay attention to what the internal or perhaps an advanced PhD young postdoc who did not know exactly how molecular biology works. So
Instead of
if there's something strange in the analysis, you have to ask the alignment. And that can be something that people can easily check. If you have done a mistake or use some different settings in your downstream biological analysis, that's harder to catch. Also, it's harder to test there because most people use default settings or their defined setting that you have to choose. Because this is very simple, but the chances of where you could mess it up are also great. For example, you have downloaded my DNA, but you forgot to lodge the right parameters for that. So if you have done something like that, then it can easily be detected. So even experienced labs, experienced people show that they have done even if it's a first step, even if it's a single step, it's probably one of the most critical steps that you have to be absolutely sure about. So with that, shall we move on to the next one? It's done.
okay. Okay, so she asked what the question mark is. That's because when in the nucleotide sequence itself, it's there a gap. For instance, it doesn't know what amino acid to code for it. So when you translate it, it shows a question mark. And there are also at times when the sequence, you know that it's either an A or a G, but you don't know which one it is. There are letters for those as well, which I don't really know. But then there are like K and L, there are some other letters which you use for such unsurety. So if that's there in the nucleotide sequence, again, when you translate it, you get a question mark because it's not sure what the amino acid should be. Yeah.
Whether you're not sure or whether there's a polymorphism. So
That
sometimes
also.
you may have is there that's when you use that, not only when
That's true.
but also when there's polymorphism.
Yeah, you had the question marks or like the if they are toward, I mean, what I do is if they're towards the end and I'm not going to miss out a lot of data, if I trim the sequence until that point, I delete it. But if there is if it's sort of in the middle and there's a lot of information that I'll be losing, and this is just one site which is we are unsure of, or there is a polymorphism, I would keep it because that is also information actually, right?
There's one more problem. Sometimes when you get your sequences, it might just happen that the sequencing machine not read that particular there's still lots of things that people cannot exactly explain. There are individuals we know where you use very, very hard markers to use markers. And then in your entire batch of individuals for that particular or whatever, you are going to get messy parts. That's the thing that happens. So when you get that, you just have the question mark that you have no idea what the problem of sequencing is going to solve that. And I have asked around with when I was doing B6 and people just said, do it again. Do it again. It's exactly the same samples. You'll again get some less there. So those kinds of sequencing errors cannot solve in the those typically coded as question marks. People didn't have the right thing out. So you could either have gaps in the beginning or end in some sequences. And again, this is more of a problem in Sanger sequencing.
not so much in a lot of more modern chain sequencing techniques. But most of the data that you will see for these individual genes in bacteria and other butterflies, which butterfly birds, certain other groups which have been sequenced for a long time. Kind of sequencing was a massive advancement, right? And that is when most people started doing this in non-model organisms like birds and butterflies. So a lot of these data sets will have missing pieces in the beginning or end. So those are just missing data, and that's because when you start sequencing in the beginning, you're going to get some crash. In the end, you're going to get some crash after your primer sequence lines. So that you simply have to just remove, or if some sequences are missing, then you just put question marks. These are various ways why you have. But again, if there are only a few, you put them as question marks. So that's missing data for those. The first column, for example, here in this data set, the first and the last samples have a question mark, meaning that you're missing data for those two, but you don't have data for the remaining ones. So it's okay if you have missing data in some parts for some samples, as long as you don't have missing data in large parts of the and for some samples. If you have it, then it's best to perhaps take out those samples which have a lot of missing data, or cut out actual the sequences where you have a lot of samples missing a lot. If this is a way to print the data set, it makes your data set smaller, but it also makes it more reliable for the rest.
Okay, so it's no. I was going to say that right now it's actually a coffee break. So there were a few people who had questions, so they can maybe ask that, while the others, this until 3:15 is coffee break. So, yes.
Those who want to go,
Yeah.
go grab your coffee, those who want to stay here and ask
So I got
questions,
stuck. And then
get things sorted out.
did the alignment. And I did align by muscle.
Yeah.
Got the translated protein sequence standard genetic code.
Hmm.
So does this look right?
No, there are too many stop codons, no?
So you can do that, and there are certain
haven't
kinds of
looked now.
analysis where you
Okay, go back to the
want to
DNA
use it.
sequences.
So especially
And
when
we'll
you're
try doing
looking
this
at,
thing
high
where,
level
like he said, your
classification,
beginnings, there
there
might be one or two
typically
nucleotides
people,
missing
do this
in some of the
either
sequences.
highly
So we try
conserved
deleting
or
one
they will,
column, one entire column from the
protein
beginning.
sequence
No, it's not the
rather
beginning.
than nucleotide sequence, because
just
proteins
click
do
that
not
column?
evolve as
I don't know
fast
if
as
Yeah,
they
click it.
nucleotide.
Nope.
So
Yeah, then
therefore,
click this
high
G and this,
categories
then
for
shift
deeper
select
spans,
the
people
bottom one.
use
No, wait.
either highly conserved
Okay.
genes with very slow evolutionary rates
Here,
and or
then
protein
here.
sequences.
What did you do? You just went that and then shift.
Won't delete. Then right click delete. Yeah.
Or
Delete. Okay, now translate it.
Why are we
No,
doing
just
that?
translate. Oh, why? This is just for protein codon genes to make sure if you have actually aligned it properly. This is just a sanity check of sorts. So if it's a protein coding gene, you should not have stop codons in the middle, which you wouldn't see once you align it. So it's just a check. Translated
how do I do
protein. No, you don't need to align again.
Yeah.
I mean, yeah, you just need to translate it again.
Right, just click on that.
Okay, so there's still some
Yeah.
stop codons. So you go back again, and then you delete one more column. Because that's good
That's
at the
not
beginning.
the.
So that's as far as it can go, right? If it's
Yeah.
no, the G.
Oh, because that
I
one's
think it
already
had
deleted.
multiple
Yeah.
G's. Shift G, delete.
And then back here and just.
There you
Oh,
go.
that looks better. Cool. Amazing. Thank you so much.
Yeah. Stop codons, those asterisk things. Go to the beginning. Translate. I mean, go back to DNA sequences. Translate the cytochrome. Yeah. So translate it. So are you using what is the genetic code that you're using to translate it? Is it the ones which is, this is a mitochondrial gene.
you say use this for the rest of the session or something? So, use those ideas.
If I have two gaps here. These two gaps. Both these so it's do I need to reverse complement it or something? And then no, don't have. It's just that approach is complement. So which do you think that it can be here you can see that. For this question. But so maybe it's a partial sequence to the the species have random thing. But if out of regions are missing, just avoid that from
So the has but okay, you know, one data set. There's also some amount of bioinformatics.
Oh, he's already there. There is one girl from University of Hyderabad. She's conference. No, not conference. She was there in my lab. It is good. Yeah. TV can. It's good. This- What would you mean? This should be for this. Not much intention. With her family. I can definitely understand that. Your parents day. Like, say, it was continuous. But it was. No, that's not a bias.